Okay, so getting back to that chemical breakdown, and this will probably be a little bit challenging, like regulation is challenging, because we're really talking about a bunch of different chemicals that we collectively call enzymes, and how they manage to break apart bonds, and what is produced from that. So these proteolytic, which means protein lysis is breaking apart, so something that breaks apart proteins themselves. The enzymes that break apart proteins are produced inactive. Now, the reasons they are inactive is because, well, most of your own cells in your body are made of the same proteins, the same proteins that you find in your food, right? Your food is made of cells from different animals, but we have very, very similar proteins. So the enzymes you produce in trying to break down proteins from the food you eat, the enzymes wouldn't know the difference between the food and you. So you have to protect you, right? And one of the ways you protect your own cells made of proteins from the enzymes that you are using here in the digestive tract is to make those enzymes inactive, so they are not able. You saw this when we talked about pepsinogen. So you keep them inactive, and that way they won't just start digesting your own cells. They'll wait to be activated in the lumen of the structure or in the space in the structure, and that will minimize the damage to you. All right. So here we say these enzymes made by the pancreas, called proteolytic enzymes, are inactive. They will become activated in the duodenum and in the lumen of the duodenum by a specific enzyme known as enteropeptidase, sometimes called enterokinase. And so this is actually a duodenal or absorptive cell enzyme, so do bear that in mind. The enzymes made by the pancreas, which digest proteins, are trypsinogen, right, procarboxypeptidase, and chemotrypsinogen. I know, words I'm sure you've used commonly in your language, right? So, in your everyday language. So no, it's not. So they're going to, it's going to be a matter of spending a little time thinking about, these words and trying to match them to function. So trypsinogen becomes activated to trypsin when it is exposed to enteropeptidase. All right. Once trypsin is activated, it can activate procarboxypeptidase to carboxypeptidase and chemotrypsinogen to chemotrypsin. And all three of these, right, proteolytic enzymes made by the pancreas are powerful enzymes. Also made by the pancreas, the pancreatic amylase. We haven't done a whole lot with carbohydrates up till now, right? We produce salivary amylase in the oral cavity. It worked for a little while in the stomach, and then we just haven't done anything else. So this amylase will make sure that the carbohydrates we have in our diet become disaccharides. And our disaccharides are lactose and maltose and sucrose. Pancreatic lipase is made by the pancreas, of course, and this will take over from that lingual lipase, and it works and does the rest of the digestion of our fats, which are called triglycerides, for the 3 fatty acids and the glycerol. And it will make sure that these triglycerides become monoglycerides and 3 fatty acids. Nucleases, right, will work on the RNA and DNA. As I said, you are consuming cells, and cells have nuclei, and nuclei have nucleic acids. And those are useful building blocks for you as well. So the pancreas makes chemicals called nucleases, and you break down nucleic acids to nucleotides. All right? And I say that these enzymes are active in the pancreas, but they tend to work or are optimized by the conditions in the small intestine. And so the lipases would require the bile, absolutely, to be most effective. All right? So secretions, yes, one more time, controlled, so that you are appropriately making and releasing these compounds from the pancreas when there is food, right? And substrates for them to work on. So the neural control is a parasympathetic, right? Hormonal is secretin and cholecystokinin, which we've mentioned already. And again, secretin release when acidic chyme comes in, and secretin really increases this manufacturing release of bicarbonate from the pancreas. Cholecystokinin release when fats and proteins enter the duodenum, and it is very much responsible for causing, right, the release of those of trypsin and chymotrypsin, carboxypeptidase, amylases, etc. So, all right, the gallbladder is where we've stored the bile that the liver made. So this is also an accessory. Bile is formed by the hepatocytes, transferred for storage to this structure. So here's the ducts that eventually, from the triads, you'd have a bile duct coming off of each one, and they'd all converge finally into major ducts from the right and left lobes, called the right and left hepatic ducts. And there'd be a common hepatic duct that then would move bile into the cystic duct and store it in the gallbladder. Right? And then the gallbladder is going to release that bile when there is a signal for it. Thank you. Sorry, tired of dogs. Okay, so the muscular sac of the gallbladder with the cystic duct joins, again, the common hepatic duct to form the common bile duct. And this will lead down into the duodenum. Bile is released when fatty chyme enters the duodenum and it is triggered by cholecystokinin. All right. So we've gotten all the structures and secretions noted here. So we want to look at this idea of getting proteins to amino acids, getting carbohydrates to monosaccharides, getting triglycerides to fatty acids and monoglycerides, getting nucleic acids to nucleotides, bringing in vitamins, bringing in minerals, and recovering all this water that you ingest and water that's been secreted by the glands and structures. So your absorption is really going to happen here. So again, this is a nice cartoon that goes back through and says, okay, here's all the parts in their entirety, and here's what happens, right? So we are to come back to this if you're lost at any point, right? You consume large molecules which have to move through the cells to get into the blood, and this process is called absorption. So those large molecules have to become smaller, as I've said, and they become smaller by chemical digestion. So looking at the carbohydrates, again, recapping this, we take in long, large molecules known as carbohydrates. Our average diet is about half carbohydrates. Less of the simple, hopefully, disaccharides and monosaccharides. Right? This begins in the mouth with salivary amylase. Salivary amylase continues to work a little bit in the stomach, but it is eventually destroyed. So carbohydrate digestion continues with pancreatic amylase, which converts, right, the substances, the carbohydrates, to disaccharides, right, maltose, sucrose, and lactose. Now, it turns out that the cells, the brush border absorptive cells in the small intestine, have these enzymes called disaccharidases, and they're shown here, maltase, sucrase, and lactase. And these three will convert all the carbohydrates that you've ingested to glucose, fructose, and galactose. Those are your monosaccharides. These are small enough to be absorbed. For proteins, again, consider we didn't do anything in the mouth or anywhere all the way down to the esophagus. In the stomach, we started with pepsin. Proteins overall, about 15 to 20% of your diet. So you have to break them down small enough to get them into the blood. So pepsin, which works at the very low pH, will start the breakdown of your proteins. It's aided by hydrochloric acid. And then the food stuff will move on with partially digested proteins into the small intestine. And in the small intestine, trypsin, chymotrypsin, carboxypeptidase will continue this breakdown and will now have dipeptides and tripeptides. So very small chains of amino acids, but still not small enough. So again, here the brush border cells, those absorptive cells that make up the mucosa of the small intestine, make these enzymes aminopeptidase and dipeptidase. And these then, right, produce, cleave the final bonds so that we only have amino acids. So all proteins that you ingest become amino acids through this process. Right? Lipids and nucleic acids. Lipids should be less than 30% of your diet, and they are mostly triglycerides, and we suggest, again, unsaturated fats as well. Lipases, you had a lingual, and we didn't mention much about this, but because it's not doesn't do a lot of digestion for us, but there's a lipase made by the tongue and by the stomach. But they really work on very special short-chain fatty acids. Most of your dietary fats are long-chain, and so it's really the pancreatic lipases that do most of the breakdown of your dietary fats. And this, of course, is absolutely assisted. Absolutely, it's invaluable to the working of lipases is bile. So bile is really, really important to get pancreatic lipases to work. And bile emulsifies, right, so forms these droplets that we call micelles. And once the droplets are formed, then pancreatic lipase is a whole lot more effective. Nucleic acids are going to be in any cells you eat, because I said your cells will have a nuclei, so yeah, you're going to find lots of potential nucleic acids. Pancreas makes these nucleases, and then brush border helps as well with that, so that we can absorb nucleotides. I say now you are small enough for the absorption process, so that was recapping the digestive from beginning to end. You use the same transports that you used back that we first mentioned to you in AMP1: active transport, passive diffusion, facilitated diffusion, co-transport, and even endocytosis, really large things. And this, of course, walks you through all the volumes of fluids that have been dumped into your tract, and the fact that you have to recover, right, these things in this process we call absorption. So since the cell membranes are phospholipids and they are water soluble, right, the phospholipids are going to interact with water soluble nutrients, but will not let them pass through the fatty acids, you have to use transporters. So phospholipids are going to interact but not allow transfer of the water soluble nutrients, which are your amino acids and your monosaccharides and your nucleotides. So you have to have transporters for them. And I note here too that these absorptive cells have very tight junctions between the cells. So in other words, nothing that you have in the lumen of this tube is just going to slip through. Lipid soluble nutrients will diffuse. So your fats, right, your fatty acids and monoglycerides are simply going to diffuse into the blood, well, into the lymph, right? So carbohydrates and proteins, right, water soluble, so they will be actively transported into the absorptive cells, right? So carbohydrates, monosaccharides is the form they're going to be in now, glucose, galactose by co-transport, fructose by facilitated diffusion. Proteins will come in only as amino acids. There are 20 different ones, but they are all co-transported. So co-transport is, again, active transport. Lipids come in, they form micelles so here's our absorptive cells. Here's micelles. They interact with that watery chyme. In a micelle is a structure that has an outside that's hydrophilic, likes water, and inside that is hydrophobic. Right? So inside we'd see fat-soluble vitamins and long-chain fatty acids. All right? So here's the micelles formed from the emulsification process. So they show you this. All right? These micelles contact the microvilli, and the fat-soluble stuff simply diffuses, simple diffusion into the absorptive cell. Inside the absorptive cell, it forms a structure called a chylomicron. And this is the first lipoprotein that we form. And that then will enter into the lacteal because it's too big for the capillary.